Hi guys, my name is Maeve Robertson, and on behalf of my group, I'd like to talk to you today about 3D printing, specifically bioprinting. The process of 3D printing is one that's become increasingly popular in the past couple decades. This machine uses digital models created on a computer to create three-dimensional real-world objects. They're printing toys and jewelry, designer clothing, and I guess even pizza now. So while this is all exciting, scientists have gone even further. The goal now is to begin printing human organs. I know this sounds crazy, and it kind of is. So let me back up a little bit, and I'll give you some background information. After that, I'll do my best to show you how bioprinting works, so you'll understand the method behind the madness. I also want to discuss the kinds of things that they've printed so far, and what they plan on doing in the future. So before we go into detail about the process of bioprinting, I think it helps to be familiar with the history of this kind of technology in general. So let's go back about 30 years, and it all started in 1984. A man by the name of Charles Hull developed a thing called stereolithography, which is a really big word, but what it means is that he figured out a way to create tangible three-dimensional objects that could be created from digital data. And, thus, this was the first form of 3D printing. 12 years later, Dr. Gabor Forgax and his colleagues observed how cells stick together during embryonic development, and they noticed how the cells move together in clumps, which seemed to have liquid-like properties. Then, in 2003, Thomas Boland, a researcher at Clemson University, was able to modify an inkjet printer so that it could accommodate and dispense cells in scaffolds. A year later, Dr. Gabor Forgax comes into the picture, once again, and this time developing a new technology to engineer 3D tissue with only cells this time, without the use of scaffolds. The leader in the field of bioprinting today is a California-based startup organization titled Organovo. This company was set up by a research group from the University of Missouri and was actually led by Dr. Gabor Forgax. In 2009, Organovo created the first ever bioprinter. They finally did it, and it was called the NovoGen MMX bioprinter. And to do this, they used Forgax technology. Then in 2010, Organovo created the first blood vessels to be bioprinted, using cells cultured from a single person. Now, in more recent developments, Organovo has succeeded in developing multiple drug discovery platforms, such as 3D bioprinted disease models made from human cells. So there you go. Now you have a brief history of bioprinting, and now we can get into the fun stuff. How do you make an organ? The process of bioprinting is, as you can imagine, a complicated one, so bear with me as I try to explain this to you. First, let's gather our materials. You'll need human cells, which can come from a biopsy from the patient, or you can use stem cells. Next, you'll need hydrogel. Hydrogel is essentially a network of polymer chains that are hydrophilic. This is where the cells are going to live. It's filled with nutrients, vitamins, proteins, and other important compounds that will allow the cells to sustain life. These hydrogels are highly absorbent, as they are over 90% water. Hydrogels are preferred for tissue engineering because its flexibility is actually similar to natural tissue, and this is due to the fact that it's mostly made up of water. So when you mix this with human cells, the combination mimics 3D microenvironments of normal cells. So basically, without hydrogel, the cells would likely settle and clump, and this would make them useless. And lastly, you need a bioprinter. So to review, you need human cells, you need hydrogel, and you need a bioprinter. Now, to print an organ for a specific person, measurements must be taken. You need to know the shape of it, its size, and where it needs to be placed in the patient's body. CT or MRI scans of the organ are taken, and the images are loaded onto a computer to build a 3D blueprint, mapping out the X, Y, and Z axes of the corresponding organ. Next comes the hard part. The scientists now have to digitally build the organ slice by slice, and each slice should directly reflect how the specific cells and their surrounding matrices fit together and work together in three-dimensional space. This is extremely complex, especially when making more intricate organs like livers and kidneys, because they require multiple different types of cells. And because they require the use of many different cells, many different types of bioink must be made, which brings us to the next step, the making of bioink. Now, when I first read about bioink, I was really confused about how you could put cells in ink, but it turns out it's nothing like that, and I was thinking about it all wrong. As we have learned, cells make up tissues, and tissues make up organs. So, to print an organ for a specific person, doctors have to create bioink using the individual's own cells. You can do this in two ways. One way is to deposit cells from a biopsy that are specific to the organ that you intend on creating. So, for example, if you want to print a liver, you would need to collect hepatocytes and other supporting cells necessary for liver functioning. Simple, kind of. A second option is to start with stem cells. So after printing, the stem cells are able to rearrange themselves and differentiate into the desired target cells. And if you think about it, this is no different than cells in a human embryo instinctively knowing to make complex organs. It's crazy. These cells need to be suspended in a growth medium, aka your hydrogel, in order for them to multiply and grow. When the desired amount of cells are created, they are collected and put into the cartridge of the printer. So, this is essentially your bioink, all of the cells put together in the cartridge. And once you do all of this, file, print, and let the printer do its job. The bioprinter now prints a layer of hydrogel as the space holder for the printed tissues. Remember that this is the temporary mold that kind of holds the cells together in the early stages. And then, from a separate nozzle, bioink spheroids are deposited into the layer of hydrogel. And these steps are repeated, and as precise layers are built on top of one another, the bioink spheroids will actually fuse together naturally. But the process doesn't just end there. The printed tissue now needs to go through a maturation phase, which takes several weeks for the cells to grow into its desired shape. During this time, the hydrogel is removed. I know that was a lot, so let's review. First, either stem cells or cells from a biopsy are cultured in order for them to grow and multiply. Next, the cells are collected to make bioink, and they are loaded into the cartridge. A separate cartridge prints a layer of hydrogel, and this is where the bioink is deposited. The cells are layered on top of one another, and eventually the spheroids of bioink naturally fuse together. The printed tissue is left to grow, and the hydrogel is removed. Congratulations! Your organ is now ready to be used. So I guess the next question to ask now is, how do they plan on using these printed organs? Earlier in the video, I know I gave you a brief overview of some of the things that they have been printing with this technology, like Organovo's success with printing blood vessels, but doctors from different parts of the world have made equally impressive contributions as well. In 2012, doctors and engineers in the Netherlands used a 3D printer made by the company called Layerwise to print a customized three-dimensional prosthetic lower jaw, which was implanted into an 83-year-old woman suffering from a chronic bone infection. This technology is currently being explored to promote the growth of new bone tissue. Then, in 2013, researchers from the University of Edinburgh created a valve-based cell printer that spits out human embryonic stem cells. This is huge because this could be used to create tissue for testing drugs or growing replacement organs in the future. Then, in a study reported in a September 2010 journal on tissue engineering, a man named Lothar Koch from the Laser Center of Hanover in Germany and his colleagues were able to laser print skin cells. This finding was able to demonstrate that their technique was suitable for computer-controlled positioning of different cell types without damaging the cells and their genetic makeup. In the future, they hope to use this application in the generation of tissue replacement in burn victims, especially in the case of extensive burns. The same group from the Laser Center of Hanover used laser printing to create grafts from stem cells that could develop into bone and cartilage. As I stated before, Organovo is currently the top competitor in the field of bioprinting. They are now creating tissues that are made available through testing services, customized projects, partnerships, and eventually for therapeutic applications. Currently, the company is building 3D tissue models for research and drug discovery experiments. So they basically can sell drug companies and laboratories their printed tissues for them to use in tests. They are now offering liver testing services, and these tissues are titled the X5 3D liver, which can be used for acute and chronic toxicity and metabolism studies. These liver tissues can stay fully functioning for up to 40 days. Similar to biopsies of human tissues, different types of tissue damage can be examined in this printed liver tissue using various types of treatments. The X5 3D liver offers precise and reproducible architecture, and it is used to assess liver tissue-specific toxicity markers. Bioprinting has also benefited the development and study of new medical research and drug testing. By using 3D printed tissues, potential treatments can be observed in real time. This allows them to study the safety, efficacy, and mechanism of action of different kinds of drugs in printed tissue samples. This technology is now being used for the generation and transplantation of several kinds of tissues. These include multilayered skin, bone, vascular grafts, tracheal splints, heart tissue, and cartilaginous structures. And here you can see a tracheal splint that they developed. There are so many things that they're talking about doing with this bioprinting technology, but the long-term goal of bioprinting is to build human organs for surgical therapy and transplants. This could benefit the healthcare system in a number of ways. For example, it will be able to decrease the time patients must wait for an organ. And because the doctors will use the patient's own cells, it decreases the likelihood that the body will reject the transplanted organ. If this all happens, it will eliminate the need for organ donors in general. And another potential benefit is that by printing human tissues, this might mean that researchers will no longer need to perform tests on animals. However, much work still needs to be done before all of this can happen. Significant developments in science are required to develop tissue therapy. More controlled clinical trials need to be performed to ensure the safety and efficacy of this technology, and the developers still need to gain regulatory clearance to use bioprinters as a medical device. Another thing to consider is how bioprinting could affect the drug industry. On average, it costs $1.2 billion to develop a new drug, and it takes 12 years to develop it. Currently, $50 billion are spent on research and development each year, and this produces on average about 20 new drugs that are approved by the FDA. The use of bioprinting in drug research could potentially impact the speed, predictability, and cost of successful drug discoveries in the future. As with any new advances in science and technology, there are a number of ethical concerns that must be addressed. Despite all the good things that bioprinting could do, one could argue that this whole concept is like playing God. There's always the argument that this is simply not natural, and that this goes above and beyond the type of control that we should have over our bodies. Moreover, it raises issues regarding population control. If we can end the shortage of organ and tissue donations, people will be living longer, and then we run into the issue of whether we may be creating bionic humans, so to speak. The idea of 3D printing organs is controversial on its own, but it also employs the long-time controversy of using stem cells for research, which can upset people on religious and ethical levels. Here's another thing to consider. What happens when complex, enhanced organs involving non-human cells are made? Who will control the ability to produce them? And how will they be able to ensure the quality of these resulting organs? Of course, one would hope that bioprinting would only be used for the best intentions, but this may not always be the case. People can turn and use this technology negatively. Similarly, once this type of knowledge becomes common, it may be produced with inferior quality. This could lead to dangerous health issues and could create a potential black market for human organs. Just as people travel to third world countries for cheaper plastic surgery, this could very well happen with bioprinted organs as well. Lastly, when these types of procedures do become more available, they will be very expensive. Therefore, they will only be available to the wealthiest customers. Many people believe that the distribution of wealth in our country is already flawed, and if only the rich can afford these procedures, those with a low socioeconomic status will be left without the money to afford keeping themselves and their families alive. This could cause resentment and even more social issues regarding wealth. Despite these potential drawbacks, scientists truly believe that bioprinting, when used for the right reasons, can have major positive impacts in the field of medicine. It'll be exciting to see what they come up with in the future, as the possibilities are virtually endless. So that wraps up our discussion on bioprinting. We hope you enjoyed watching and that you learn more about this new scientific revolution.